The first speaker today, starting this session, which I really hope you like, is Giorgio Colombo from the University of Pavia. And the title of his talk is The Allosteric Code of Molecular Chaperones from Mechanism to Drug Design. Please.
So thank you, Giovanni. I'd like to thank you very much and all the organizers and also this ALLODD network for giving me the chance to be here again in Urbino. I've been here before as a tourist, but I couldn't remember, it was so beautiful. So yeah, it was really great to come here today and yesterday. So what I'm going to tell you about is what we have done in the last few years on using molecular simulations and computational approaches to try and develop systems to understand the complex mechanisms of large molecular chaperones, and in particular the chaperones of the HSP90 family. So as you see here, basically these chaperones undergo large conformational changes which are triggered by the binding of a nucleotide, ATP, which send a conformational signal through the structure of the paper of the protein to a distal site of the protein that triggers the opening and closing of this large molecular ligand. And this gives the timing for the chaperoning cycle of the whole protein that has to go through a series of conformational changes to recognize substrates that it will fold correctly in the three-dimensional state and release also the substrates. And the substrates are called client proteins. So, as I said, the chaperones of the HSP90 family are regulated by ATP or ADP. ATP is hydrolyzed to ADP, and then something happens in the structure of the protein. And what we wanted to investigate is to try and understand where the signal ends up and whether the sites where the signal ends up can be targeted by small drugs or small molecules that then will send the signal back up, regulating how ATP is processed and the whole function of the chaperone. And we became particularly interested in that because there are some cases where these chaperones, HSP90 in particular, are particularly overexpressed in some types of tumors. And people have tried to inhibit HSP90 chaperones by competing out the nucleotide from the active site. And in particular, they have tried to compete out ATP by small molecules, but ATP binds to a site that is highly conserved among all the isoforms of the protein. And some of these isoforms are still needed by the cells that carry out normal functions, non-cancerous cells. Other types of isoforms, and in particular the mitochondrial one that you can see here, are in some types of tumors highly overexpressed. But of course, if we then compete out the nucleotide by using molecules which will mimic ATP, we will target the whole family. So we were interested in targeting the allosteric site to provide an opportunity for having some selectivity, or as a means to have selectivity among the different chaperones. And in particular, we know that allosteric sites are less conserved and also structurally may be different among different proteins of the same family. And as you can see here, even in the X-ray structure of the cytosolic version of the protein versus the mitochondrial version of the protein, you already immediately see that you have some differences in the sense that the mitochondrial protein TRAP1 crystallizes in an asymmetric form. So we asked the question, is there a difference between the way that the message from ATP is relayed through the structure of ATP, of HSP90 or TRAP1? Do they target different sites? And to do that, we developed a very easy computational approach where we just try to calculate how residues are coordinated by looking at the distance fluctuations in every pair of residues in the protein structure. So we take residue I and residue J and look at the fluctuation in their distance along different simulations and compare them in different cases. So if we have two residues which are highly connected, you may imagine that they behave like the ends of a rigid body, which whereby the distance fluctuation is small, and so I and J, which would maybe the tips of my fingers here, will move together consistently. So if I perturb here at this point, the perturbation will be mechanically relayed down to the other point. If two residues fluctuate a lot with respect to one another, then the information encoded here will be kind of lost along the way to the other residue, right? So they function like this. So by using this type of simple analysis that we have developed over the years, we can check the workings of the protein in different nucleotide states. And in particular, we looked at the 2-ATP state, and mixed nucleotide states. And we report here in this figure only as red dots the residues which are highly correlated, highly coordinated to the residues of the active site containing ATP in the different simulations. And what's interesting here is that the residues that are always highly coordinated to the ATP site always involve some sort of some residues here in this middle domain, which are also the residues that are mostly involved in the conformational transition that the chaperone has to support when it folds client proteins. It has been seen by other techniques, including cryo and thread and so on by the Regatta. So here, what we may imagine is to have a possible site where we can, which we can target by small molecules. And so what we did was to scan this site by pocket finding algorithms to find pockets to host small molecules. But one thing that is important to show here, to underline here, is that we have a different organization of the putative allosteric site in TRAP1, the mitochondrial part of the protein, which is here on your right, versus the cytosolic putative allosteric site. In one case, one is completely asymmetric. In the other case, the putative site is completely symmetric. So we scanned the site on TRAP1 with these pocket finding algorithms, SiteMap, and others to obtain a consensus, and then we built a pharmacophore model complementary to the functionalities presented by the amino acids in the identified site. And we use this pharmacophore model to screen for small molecules, fragments, or drug-like molecules in databases that had them available to purchase. And then, in collaboration with our collaborator, Andrea Rasola at the University of Padua, who's an expert in mitochondrial biology, we tested some of these molecules against TRAP1 and also against the cytosolic form, which is HSP90. And what you can see here is that while normal inhibitors, in particular, like the vancomycin and radical, which target the ATP site and are known to target the ATP site, inhibit both proteins at the same level, the small molecules selected for TRAP1 are actually inhibiting TRAP1 to a larger degree than they inhibit HSP90, in some cases with even opposite effects. And in particular, there's a series of 3 compounds that have high inhibition of ATPase rate and also of ATPase turnover of this protein. Then we tested these molecules in the case in cancer cell Foci formation, and here you see the formation of Foci of cancer and the treatment of the Foci with different amounts of one of the small molecules, which is compound 5, and you see that the Foci can become abrogated in the same way as they are abrogated by knocking out TRAP1 using CRISPR-Cas9 approaches. So the small molecules, in a way, phenocopy what happens at the level of CRISPR-Cas knocking out, indicating that we are actually at least targeting TRAP1 and inhibiting some of its cancer or carcinogenic activities. So this gave us a series of compounds that are really interesting as allosteric inhibitors of TRAP1, selective isoform selective allosteric inhibitors of TRAP1. But then we also became interested in other types of forms and functions of this protein. And in particular, it is known that there are some forms of the protein with again, one more name, GRP94, which is the endoplasmatic reticulum form, which is known to become highly glycosylated when you are in a disease state, when cells are in a stressed state. And cancer cells are, of course, highly stressed because they have to survive in the face of an environment that wants to get rid of them. So they have to turn on their chaperone machinery, their defense machinery, their heat shock response machinery a lot. So it came out from the Chiosi's lab. MSK see that these chaperones, this family of chaperones, is highly glycosylated in these states. There's in particular, two types of glycosylation that transform this protein from a protein that folds other proteins from a protein that holds other proteins there and transports them to the plasma membrane and helps them survive when they are partially folded or partially unfolded. And so the question that the Chiosi's lab asked was, how do the sugar, is there a way that we can model the fact that these sugars on these two particular locations impact on the function of the protein, and can we sort of use this to rationalize this functioning? Is there some allostery involved, and can we use this information then, if we get to a decent model, to design molecules that may have function solely against this glycosylated form? So we started running simulations of the glycosylated form of the protein, comparing them to the non-glycosylated form of the protein. And we did it with different glycosylation states. So we used the fully glycosylated version of the protein with glycosylation on residues 217 and 62, the monoglycosylated form, and on one amino acid only, and the other monoglycosylated form. And we have data from the Chiosi's lab for all of this. And the pathologic one is the N62 glycosylation, the one that is really related to pathology. So what you can see here is that from long simulations, what we did was to refit all the structures of the protein on one reference structure and let the sugars move freely in solution. And what you can see in this sort of hairy representation, hairball representation, is that mostly the sugars tend to be out in solution and to be sort of disordered. But in particular, the ones related to the presence of, to the position, which is known to bring glycosylation to disease effect, extend their activity to form interactions with the long loop, which is present on HSP92, and these chaperones to bring signals from this region here down to the C-terminal domain, while the other sugar doesn't do that. This is an exclusive effect of the sugar on position 62. So in a way, hyperglycosylation covers the active site domain. It may impede ATP to enter, but it is important to notice that even though it uses different combination of low energy interactions, the glycosylations play different roles, and they depend on the positions they are located. So to delve more into this, we also looked at how the glycosides may change the way that the lead closing over the ATP to form the catalytically competent state is regulated. So you may imagine that here in the active site, you have a lead that covers ATP and needs to cover it to have catalysis. So we can define a closed state, which is the catalytically competent one, and an open state, which is the catalytically non-competent one. So when we look at the completely glycosylated protein, we always see that glycosyl at position 62 tends to reach out to the ATP lead and pull it open completely, so that ATP can either enter and exit very quickly or be released also very quickly. When we have glycosylation only on the carcinogenic position, on position 62, and we can do that by mutating out to residue 217 to an alanine, then again we observe the same effect, and the open lead is favored, although to a slightly smaller effect. But when we then try and chop off the glycosylation at position 62, then the lead closes up. The majority of conformations of the lead are closed, and even more so when no glycosylation is present on the. So the idea here is that glycosylation not only changes the dynamics of the protein by touching the long loop that connects two domains, but also fine-tunes the opening and closing motions of the catalytic site. And this, of course, will change the way the whole protein works throughout the chaperone cycle. And then the next question is, okay, but then the lead, we know, is sort of a sensor for the nucleotide state that then gets relayed and then it sends information to the client binding site. Is there an effect of the sugars that goes far to the client binding site, to the protein substrate binding site? And we looked into that again with the tools that we developed before, of course. And we noticed that in the case of the wild type ATP state, so the functional one with no sugars on, the client binding site down here is very flexible, can adapt to the client binds and gets released. It shows more opening than the case where two glycosyls are present. The site is a lot less flexible and a lot more closed, so it really will hang on to the protein that has to be folded and keep it there and perturb or change its function. One more thing that came out of this simulation is that we have different conformations that are selected by the sugars. So can we use these different conformations that are selected by the sugars to design, to rationalize the effect of small molecules that we know, or the Chiosi's lab showed, that can target one form of the protein versus the other? And so to do that, we thought that, we may have a selection in terms of the fact that as the lead is more open, possible ligand that gets into the site should stay there longer to be active, while a ligand that is not active should probably be released more easily. And so, thanks to Sergio, also who's here in the audience, and Vicky that gave us a great hand in this case, we tried to calculate the unbinding times of different sets of compounds, active versus non-active, from different conformations selected from the sugar or non-sugar bound structures. So in the case of the sugar-bound structures, we have this lead that is partially open, and the active drugs always have a longer residence time than the non-active ones. Then we extended it to another series of congeneric analogs and showed that systematically the more active drugs tend to bind to the semi-open or to the open state of the lead with a lower unbinding times, with a slower unbinding time than non-active ones. So meaning that they should stay there more, affect their and impose their activity by staying there longer. So to wrap up on this part, the sugars do have an active role in modifying the way the active site is organized. They change through this the way that the client binding site or the substrate binding site and distal site is organized and dynamically responds to the presence of a client. And also, finally, this analysis of the sugars give us an idea of how to select or design or help design small molecules that are conformationally sensitive for the glycosyl versus the non-glycosyl part. As a final couple of slides, if I have time, I will just show you the fact that we are actually looking at one protein, which is indeed part of a very much more complex cycle where different complexes have to form, to disassemble, to form again, and so on. And this delicate balance between the different proteins can also be modulated by post-translational modifications. So we started to look into the effects of post-translational modifications other than sugar modifications on other proteins that make part, that build up these large complexes. And in particular, we looked at the one of the co-chaperones, one of the ancillary proteins that are needed to bring all things together. And in particular, this chaperone, which is called co-chaperone, which is called HOP, is known to be phosphorylated at one tyrosine. And this phosphorylation changes the way that the complexes are assembled and the substrate proteins are maturated in the cells. So we looked at with simulations at the phosphorylated version of the co-chaperone versus the non-phosphorylated one, and compare the conformational populations that are formed, are selected, showing that while the wild type tends to be mostly in the open conformation, the phosphorylated one tends to be more closed, tends to populate more this sort of enzyme. Then we tried to, it's impossible to phosphorylate selectively one of the residues in cell or but we can have mutations that mimic phosphorylation. So we designed a couple of mutations using aspartate and glutamate to mimic phosphate, so phosphomimic mutations, and show that at least in the case of aspartate, the phosphomimic mutation really recapitulates what the phosphorylation does in terms of selection of the complexes and of the conformations that are presented by the co-chaperone to form the complex with the whole chaperone complex. And then what we did was to use this information and realize mutants of the co-chaperone to test them in cell experiments where the maturation of substrates could be followed. This was in collaboration with the PCAP lab in Geneva. And what we could show is that in the presence of phosphorylation or of the phosphomimic mutations, the co-chaperone, which goes from an open conformation that binds to a closed conformation that apparently should not bind, actually do bind a lot less amount of chaperones than the wild types. And also, this has an impact on the maturation of glucocorticoid receptor and kinases, which are the substrates that have to be folded and released to be properly functioning if the whole complex machinery is working properly. So the overall message is that phosphorylation or phosphomimic mutations help select different conformations that are functionally efficient or function less optimally in the assembly of the complexes, and the effect of the phosphorylation is to modulate this timing of formation of the complexes and then of the maturation of the substrates. And finally, I hope I could show you that structure-based dynamics can be used to different extents to rationalize and design small molecules, to look at the effect of protein post-translational modifications, which are becoming more and more relevant today. And also, we can use this to drive the development of small molecules, drive mutations, drive different types of study in studies in complex mechanisms. So before continue, I would like to thank all the people that worked in these projects over the years, and also our collaborators and funding. And I'd like to thank you all for your attention.
Thank you, Giorgio. That was super interesting. I think we have time for one question. Please.
So going back to the role of ligands in allosteric response, so I can notice, let's say, a trade-off between the conformation freedom of ligands and the reduction or increase of conformational freedom of the protein. So,
yeah, so there's a combination of two effects. So I wouldn't talk about reduction of conformational freedom of the glycans, rather than the glycans in specific positions can establish a series of interactions, many of them, each of them pretty weak, that select one of the conformations that are consistent or compatible with one of the conformations. So it's a combination of two flexibilities that become selection for a certain function.
All right, so I think we can thank Giorgio again.